Imagine the searing pain of a burning stove, the sudden shock of ice water, or the gentle weight of a loved one's hand. For thousands of years, the exact way our bodies perceive these invisible forces remained nature's most impenetrable mystery. In the 17th century, the philosopher René Descartes imagined that microscopic particles of fire tugged on a physical thread connecting the skin directly to the brain. By the 20th century, science had mapped the gross anatomical nervous system. We knew that electrical action potentials raced up the spine, and that different nerves fired specifically for heat, cold, and pressure. But the actual microscopic gateways, the molecular machines translating the physical universe into biological thought, remained invisible. They were ghosts haunting modern neuroscience. In the late 1990s, the relentless hunt for these sensors took a radical turn. The weapon of choice was not a highly complex synthetic chemical, but the fierce, burning sting of the chili pepper. David Julius hypothesized that capsaicin, the chemical responsible for a chili's burn, held the ultimate key to understanding human pain. Finding this microscopic receptor was a monumental genetic gamble. Julius and his team extracted a massive library of DNA fragments from the sensory neurons of rodents. They introduced these fragments, batch by agonizing batch, into laboratory cells that were completely blind to the pepper's burn. They waited for a reaction. Nothing happened. Millions of cells remained biologically numb. But then, out of the darkness, a breakthrough. A single isolated gene clone suddenly brought the cells to life. They had found an unknown ion channel, soon named TRPV1. The true cinematic revelation was yet to come. When Julius raised the ambient laboratory temperature past 40 degrees Celsius, the exact physiological threshold where human warmth shifts into searing pain, this microscopic gateway blasted wide open. This was not a biological quirk for spicy food. It was humanity's internal fire alarm.
The biological door to thermosensation was blown open, and the scientific race accelerated. Julius, alongside another brilliant investigator named Ardem Patapoutian, turned to the exact opposite extreme. They used menthol, the natural compound that creates a refreshing cooling sensation, to hunt for the biological thermometer for cold. Both laboratories struck gold simultaneously, discovering a closely related receptor called TRPM8. Activated at the
precise temperatures humans perceive as innocuous cold. The complex puzzle pieces rapidly fell into place, revealing human skin to be a highly intricate mosaic of thermal antennas. Receptors like TRPA1, TRPM3, and TRPM2 were uncovered, working in a beautifully coordinated electrical symphony. To feel the gentle warmth of a summer breeze, the body simultaneously activates warm-sensing fibers and deliberately suppresses the cold-sensing TRPM8 gateways. Yet the absolute most elusive enigma still lurked in the dark. How do we feel physical pressure? How does the sheer weight of the world physically trigger a spark in the brain? This was the holy grail of sensory biology. Ardem Patapoutian fundamentally changed the rules of the hunt. He discovered a specific laboratory cell line, Neuro2A, that naturally twitched with a tiny electrical current whenever it was physically prodded with a microscopic glass pipette. Painstakingly mapped its genetic makeup and isolated exactly 72 candidate genes that might harbor this mechanical sixth sense. What followed was a highly tense, methodical countdown. His team silenced the genes one by one using RNA interference. Gene 1, they poked the cell membrane. It still felt the pressure. Gene 50, the spark remained. The tension mounted as the list rapidly shrank. Finally, they reached the absolute final candidate on their entire list, Gene 72, a mysterious sequence known only as FAM38A. They silenced it. They pushed against the cell. Complete silence. The electrical spark was entirely gone. Patapoutian had captured the ghost. He named the massive new protein PIEZO1 after the Greek word for pressure, and immediately discovered its closely related sibling, PIEZO2. These were the exact primary molecular transducers required for human touch. Their biological role was infinitely more profound. PIEZO2 was proven to be the engine of proprioception, our silent, continuous sense of where our limbs exist in physical space, the vital internal map allowing us to coordinate our complex bodies without collapsing. These proteins were utterly alien compared to any previously known ion channels.
They were staggering molecular structures composed of 2,500 amino acids. They organically form giant three-bladed propellers that curve outward, creating microscopic nanobolts firmly embedded in the cell membrane.
When a physical force pushes against the membrane, these giant blades violently flatten out. This extreme physical distortion rips open a central pore, instantly flooding the nerve with a powerful electrical signal. The devastating power of these cellular mechanosensors extends deep inside the most vital organs of the human body. They act as critical stretch receptors in the lungs, triggering reflexes that prevent fatal overinflation with every breath. They also monitor the force of blood flow, helping maintain blood pressure and prevent cardiovascular collapse. The importance of these systems becomes painfully clear in individuals born with mutations in PIEZO2. In this condition, known as PIEZO2 deficiency syndrome, patients live profoundly disconnected from the physical world. Without proprioception or light touch, movement becomes uncoordinated, limbs may develop contractures, and even basic breathing can become a struggle. The discoveries of David Julius and Ardem Patapoutian resolved a question that had persisted since the earliest days of philosophy. They mapped the molecular architecture behind how the human body connects to reality itself. For these groundbreaking discoveries, they were awarded the 2021 Nobel Prize in Physiology or Medicine. Their work revealed the molecular mechanisms that allow humans to perceive heat, cold, and mechanical force, solving one of biology's most fundamental mysteries. They showed how a microscopic particle of heat, a drop of cold, and the pressure of the physical world are transformed into the electrical symphony of human experience.